Well, welcome, everyone. Welcome to the 2013 E. coli webinar. It's titled, Beef Safety, Are You Giving Good Advice? This is the third in a series of webinars designed to communicate pre-harvest food safety to different audiences. So with this webinar, we hope to reach those who share knowledge about food safety to consumers. And we want to help you, first of all, to understand how people acquire E. coli O157H7 infections. We want to help dispel some of the perpetuated myths about E. coli on-farm occurrence and help you learn a little bit about what's known about controlling this pathogen throughout the food supply. And we hope to improve your ability to communicate the complex science associated with the Shiga toxin-producing E. coli. We have an in-house audience, thanks to all of you for being here, and as well as many people from across the country that are signed in to our online webinar. So thanks to all of you for being here. We'll take time after each speaker to take questions. Those of you attending in person here at Mississippi State University College of Veterinary Medicine, you can write your questions on the 3x5 cards that are in your packets and send them to your left, and we'll collect them. Try to do that as you think of the questions so that we have time to answer those questions. Those of you that are attending the live webinar, please send your questions to this email address, vetextension@vetmed.wsu.edu. This webinar is brought to you at no charge because of a grant from the U.S. Department of Agriculture, National Institute of Food and Agriculture, AFRI Competitive Grants Program. Dr. Thomas Besser, who you're going to hear from in just a second, is the project director for this grant. Drs. Dale Moore and myself are co-PIs serving this extension component. So with that, we'll get started. Our first speaker is Dr. Tom Besser. Dr. Besser is a professor in the Department of Veterinary Microbiology and Pathology at Washington State University. His research focuses on agents that cause human foodborne diseases, including the topic for today, which is the Shiga toxin-producing E. coli. Dr. Besser has a long history studying this organism and how to reduce its presence in livestock, and I'm very proud to be able to introduce Dr. Tom Besser to you today. Dr. Besser.
Thank you very much, Dr. Smith. It's a pleasure to be here today and talking about one of my favorite subjects. My title is a little dramatic, Carrier Cows, Menacing Microbes in Human Health, but that really is what we're going to be talking about today. But what it is really I'm going to try to do is introduce pre-harvest food safety. What is pre-harvest food safety and how do we apply it to Shiga toxigenic E. coli, which is what STEC stands for. So pre-harvest food safety is actually an old concept. It's how do we control human diseases that humans acquire from animals by interventions at the animal level. If we can get rid of these in the animals, we can reduce the exposure of humans, and that's the ultimate goal. You know, the classic control eradication programs in this country are those for bovine tuberculosis and brucellosis. These have been largely very successful. They're not complete, but they're very effective and have greatly reduced human health problems due to these agents in our country. The paradigm for pre-harvest food safety that I'd like to introduce here that I think might relate to Shiga toxigenic E. coli is that for Trichinella spiralis, the agent that causes trichinosis. And this disease is the reason we all overcook our pork, basically, because you can control these worms in their cysts by a high degree of cooking. But in actual fact, the risk of acquiring trichinella from pork, even if you don't overcook it anymore, is extremely low in this country because it's largely been eradicated from the swine population through pre-harvest food safety, where it was realized, among other changes, that if we just stopped feeding raw foods to the pigs, they would no longer acquire the trichinella and therefore no longer have the cyst to pass on to us. So it's food safety for our food, and that is one of the concepts that we've worked on trying to control STEC O157, which will be what I'm talking about today. So you probably already know this, but E. coli is a very common bacterial in the intestinal tracts of of all mammals and some other animal species. It's one species, but it includes many different strain types, most of which are beneficial or harmless. But a few serotypes, which is what we call these strain types of E. coli, can cause serious human disease. And the group that we call enterohemorrhagic E. coli are one of these. They cause diarrhea, they cause bloody diarrhea, and they can cause an acute renal failure syndrome called the HUS, for hemolytic uremic syndrome. And these agents have in common that they produce a toxin called shigatoxin, and that's how they got their name of STEC, shigatoxigenic E. coli. So how big of a problem is STEC-related E. coli? It's a pretty big problem. There's a lot of foodborne diarrheal disease in this country, millions and millions of cases. STEC is a relatively minor cause of that, although the disease it causes is more severe than most, but less than 5%. And if you look at the more severe foodborne illnesses that result in hospitalizations, it's over 100,000 cases each year. And STEC is responsible for almost 2% of those hospitalizations. And finally, the worst outcome of death, there are estimated to be 3,000 deaths due to foodborne illness in this country annually. And STEC is a relatively minor component of that, estimated 21 deaths, or less than 1% of the deaths related to foodborne illness. I want to talk at least briefly about the kinds of STEC, because there are a number of different E. coli serotypes that can produce this shigatoxin and have what it takes to cause this bloody diarrhea syndrome. And basically, there's one strain that stands out, and I'm going to call it O157. That's the serotype designation. And if you look at diarrheal disease, STEC O157 alone accounts for about 1/3 of all the STEC-related diarrheal disease. And the other 2/3 are due to, 6 at least, but probably 20 different serotypes of E. coli. So O157 is by far the single largest source of the problem. And as you look at hospitalizations and deaths, O157 becomes even more prominent. It accounts for about 90% of the hospitalizations due to STEC and about 95% plus of the deaths due to STEC overall. So O157 is going to be the topic for the rest of this talk, just because it's the one that's gotten the most research and it's the most important. 20 years ago, there was a terrifically big and bad outbreak of STEC O157 associated diarrhea associated with this fast food chain, especially in the western United States. And this, in many people's mind, is what first linked ground beef or hamburgers with this bacterium through the cattle operations. And that's still a linkage that's made. We're now 20 years later. My slide title needs to be updated here because it's after more than 15 years after Jack in the Box. But O157 is still a problem, and some of the other speakers today will go into that. And there are a lot of reasons why O157 is still a problem, and that's really one of the main points I want to get across today, is that it's a very complex problem to attack, and there are no simple answers. O157 is ubiquitous, found on every cattle farm, and we think most other farmland or rural lands around the country, regardless of whether there's cattle there or not, has a broad host range. Animals that are carrying this do not get ill, so you can't single them out and identify them by any kind of disease. Animals that become infected with this can shed hundreds of thousands of cells per gram, and yet to become infected only takes a handful of cells, less than 20 cells. And this bacterium for E. coli is surprisingly able to survive in environmental situations like water and feeds and so forth. Broad host range, this picture shows some of the species, but there's a lot more from which it's been reported. There'll be more about this later. Animal reservoirs, we think of cattle. The cattle are probably the most important reservoir in terms of exposure of humans. They don't have any disease with it. They have a really pronounced summer seasonal peak of infection and shedding of O157H7, and we'll talk about that quite a bit more, both myself and others later today. But when cattle get infected, it's a pretty short-lived infection, and most of them will stop shedding it within a matter of weeks, and occasional cattle might take a couple of months to get rid of the O157 infection. The fecal shedding by infected cattle ranges from very low, that need special techniques to find it, but they can also range upwards to 100,000 or 1,000,000 cells per gram, enough to infect an awful lot of other cattle or other animals or humans. Their infectious dose is very low, and young animals are more likely to be infected than older animals. Lots been made out of how the cattle industry has changed in the decades since E. coli 0157H7 has emerged, and the question is naturally whether these changes in production intensity, size of operations, density of cattle, and so on have something to do with the emergence of 0157H7 as a problem. We'll talk again more about this later today. In summary, we don't think so. No one has been able to systematically show a difference between animals that have hundreds of acres to themselves out on pasture and animals in a multi-thousand animal feedlot in terms of the percentage of them that are shedding 0157 at any given time. So to approach pre-harvest food safety for 0157H7, we start at the bottom of this slide at human infection, and we try to document what causes the human infection. And there's a whole list of sources of 0157 that have been shown to be able to actually cause human infection. And then we have to go back from that to determine where that contamination originated from. And hopefully somewhere we can break this chain along the way. I've highlighted in red here the path from infected cattle to contaminated meat to human infection, because that's known to be a very important route of one, but I want to emphasize it's not the only route. So I'm going to talk just a couple slides here about a topic of current research interest. Other than this, I'm not going to really talk about research today. This slide shows the calendar months across the bottom from January through December. And on the y-axis on the left is the percent positive pens when you sample pens of cattle to see how many of them are shedding 0157H7. And that axis relates to the bars, which are the cattle, prevalence of cattle infection. So the black bars are feedlot beef cattle, and the gray bars are dairy cattle. And I think it's pretty obvious if you just look at this that there's a relatively quite a low rate of infection of cattle, both beef and dairy cattle, in the wintertime. And it reaches a high peak July, August, September, and then rapidly drops again as you approach the winter again. And then superimposed on that is the red line, which relates to the right y-axis, which is the number of human 0157-related disease across the year average. That was from 2003, but it happens this way year after year after year. And I think you can see that there is a pretty good correlation there with the number of human cases of disease with the number of cows that are infected with 0157. And we think that's not a coincidence entirely, but in fact, that some of that human disease, maybe a substantial part of it, is acquired through exposure to products from those infected cows. So if to look at this simplistically, this shows those reported cases of human infection over a 3-year period. And you can see it's those summer months, illustrated by the sun here, that the problem occurs. And one approach to pre-harvest food safety for this agent would be to try to figure out some way of eliminating those peaks in cattle infection in the summertime. Oops. So if we could eliminate those peaks, we think we could eliminate a substantial proportion of the human disease by that change alone. So that's led to a lot of interest in what causes summertime peaks in cattle infection with 0157H7. And there's been a number of different hypotheses about this proposed in the literature. One is that there's something about the cow's physiology, so that when infected in the summertime, it's more likely to get infected, the infectious dose is lower, and it may shed higher amounts of 0157 in the summer than it does in the winter. A second hypothesis, if a cow is, the cow itself, 0157 challenge, but there might be other microbes in its gut that vary summer from winter, and those other microbes may have an inhibitory effect on 0157, so that given an equal exposure in the wintertime, those other microbes keep the 0157 down, so they prevent cattle from shedding a lot, but in the summertime, they enhance it. Or you could think of it the other way around, where there's microbes in the summer that cooperate with 0157 to cause infection. And then the third hypothesis is that cattle and cattle's GI tract bacteria are exactly the same summer and winter, and what happens in the summertime is that cattle ingest a lot more O157H7 in their food and water or from their environment than they do in the winter. So we just completed an experiment to test these three hypotheses in a cohort, a small study, a cohort of 20 cattle. And what I've showed on this slide are the results of 2 years study with this same cohort of 20 cattle. Y-axis is identical, which is the average number of E. coli cells shed per swab of the rectal colonization site. And on the x-axis across the bottom is the number of days post-challenge, and both these go from 0 to 60 days post-challenge. And what we did is we made identical challenge doses in a number of aliquots and froze them back in the lab. So all these cattle received identical challenges with O157H7. So in the first winter and the first summer, we challenged them with 100 million bacterial cells of O157H7 on day 1, and we followed what their shedding was. In the following year, we repeated that study, but with 100-fold less of a challenge, 10 million cells rather than 1,000 million cells. And you can see the two lines that look pretty generally similar on these two graphs, and they show that in both years, the animals challenged in the winter and the animals challenged in the summer had no significant differences in the amount of O157H7 that they shed. So what we're thinking now is that most of the root cause of seasonal variation is actually that cattle must be exposed in their food or water to O157 more in the summertime than they are in the winter. And we think that probably accounts for the whole bulk of the seasonal variation. If there is a slight difference, like in that first slide on the left, you'll note that it's the wintertime cattle that are shedding higher levels than the summertime. So it still refutes that seasonal variation. So I turned around my little chart that I showed earlier and put infected cattle at the bottom. What we now have to figure out is how are these cattle getting infected, what are the main routes of their infection, and how can we control that exposure. For the rest of my talk, I want to talk about fault. And fault finding is always part of controversial public issues. But a number of parties have been blamed for the E. coli problem. One party that's been blamed are cattle ranchers or cattle feeders. And it's thought that maybe some of their practices now are conducive to O157H7 shedding that they could do something about.